Assalamu alaikum, dear students. This is the third lecture of the cell and its uh, function. We will discuss the ribosome and some other organelles in this lecture. इससे पहले लेक्चर में हमने जब पार्ट वन लेक्चर था हमारा, उसमें हमने सेल थ्योरीज के बारे में बात की थी, सेल के टाइप्स के बारे में बात की थी, प्रोटोप्लाज्म और उसकी फंक्शन के बारे में बात की थी. We discussed the composition of the protoplasm. We also discussed the cell membrane and its structure, as well as we also discussed the transmembrane protein, the integral protein, peripheral protein, the monotropic, the biotropic, and the polytropic protein. ये सब के बाद in the second lecture then we discussed the membrane bounded and the non-membrane bounded structure. Specifically, we discussed the membrane bounded structures, the Golgi apparatus, the endoplasmic reticulum, and as well as we shortly discussed the lysosome as well. In this lecture, we will discuss about the ribosome, the peroxisome, the lysosome, and इसकी function भी हम यहाँ पे discuss करेंगे. So let's go and discuss the ribosome and other organ मेम्ब्रेन ऑर्गेनेलिज. हम कुछ ऑर्गेनेलिज डिस्कस करेंगे जिनकी मेम्ब्रेन बाउंडेड होंगी, जिनकी मेम्ब्रेन इर्द गिर्द होंगी, और कुछ जो है नॉन मेम्ब्रेन बाउंडेड भी हम इसमें डिस्कस करेंगे. सबसे पहले राइबोसोम. राइबोसोम जो है, दिस इज एक्चुअली द राइबो न्यूक्लियो प्रोटीन पार्टिकल्स होते हैं, एंड दे आर स्मॉल नॉन मेम्ब्रेनस ऑर्गेनेलिज which containing the RNA and proteins. The structure of RNA, that is almost 20 to 30 nanometer in size, and each structure of the ribosome, that is consists of two units, the large and small subunit. The large one, that is up 50s, and the smaller one is the 30s. Ribosome contain almost 65% of the RNA, and this RNA is known as the ribosomal RNA or the rRNA. RNA, there can be the transfer RNA, the messenger RNA, or the ribosomal RNA. So ribosomal contain RNA 65%, and the protein at 35%, which means by composition the ribosome, um, the RNA in the ribosome by composition is the maximum proportion, while protein they are less in proportion. You might find them floating, these ribosome, scatteredly here and there in the cytoplasm, and these floating ribosome they make protein that will be used inside the cell. So basically, they are the protein factories. They are the protein manufacturing sites. Other ribosome they are found on the surface, on the outer surface of the endoplasmic reticulum, which give the name of granular endoplasmic reticulum or the rough endoplasmic reticulum, as we discussed in the previous lecture, second lecture. The endoplasmic reticulum with attached ribosome is called the rough endoplasmic reticulum, also known as the granular endoplasmic reticulum. Those attached ribosomes they make proteins that will be used inside the cell, and proteins made for export out outside of the cell. So this is the large subunit, this is the small subunit, and these both will combine together to make the complete functional ribosome. A large subunit without the small subunit will be not working properly, and similarly the small without the large will also not work properly. So for their normal and proper functioning, these two should be combined together to make a complete functional ribosome. Now, why they are 50s and 30s? So, and what does the S mean here? So, we are going to discuss in this slide. Numbers are actually measures of how quickly each subunit sink to the bottom of a container of a liquid when spun in a centrifuge. It it mean uh, 50 is 30 is will um, sink in the bottom more quickly than the 50 is. The S is a measure of the rate of sedimentation and the centrifugation rather than a measure of weight. A Swedberg unit symbol S is a unit for the sedimentation rate. The sedimentation rate for a particle measure how fast the particles settle as sediments. So based on the function ribosome, there can be two types attached to the rough endoplasmic reticulum, which synthesis protein, and free floating ribosome synthesis cytoplasmic protein. Based on the side found, there can be the prokaryotic ribosomes or there can be the eukaryotic ribosome. The difference between them is the prokaryote ribosome they are 70S and the eukaryotic ribosome they are 80S. Now, what is the two processes or what are the processes which occur in the protein synthesis? So, two important processes in the protein synthesis is the transcription and the translation. And by ribosome, they are basically involved in the production of these proteins. So, these are basically the protein factories or the protein manufacturing site. Protein synthesis is caused by transcription, and the second is the translation. So, what is the transcription? The formation of messenger RNA from the DNA is called the transcription. So, that simple definition that the conversion or the uh, formation of the messenger RNA from the DNA is called the transcription. What is translation? The formation of the protein molecule is called translation according to the message, according to the sequence, according to the information that is being provided by the messenger RNA. The formation of the protein molecule occurs in the ribosome with the help of the messenger RNA and the transfer RNA. So this is actually the nucleus where the DNA is present inside the nucleolus. This is the nuclear cytoplasm. This is the cell membrane. So inside the nucleus, there is nucleolus, and inside nucleolus there is DNA. From the DNA, the RNA is made, and that process is known as the transcription, and this is known as the messenger RNA because they have the message, they have the sequence which the DNA has been provided to the messenger RNA. So then this messenger RNA they just come out of the that nucleus through the nuclear pore, and then it just reached all the ribosome, and then the production of that protein on the basis of the sequence bring by the uh, messenger RNA. And when that information or that sequence which is brought by the messenger RNA from the nucleus to this ribosome, this process is known as the translation. Amino acids are linked to one together to another at the ribosome to form the protein encoded by the messenger RNA. Now the lysosome. Lysosome, they are membrane-bounded organelles which are formed by the Golgi apparatus and are dispersed throughout the cytoplasm of the cell. Lysosome provide an intracellular digestive system that allows the cell to digest and remove unwanted substances. They have a number of digestive enzymes which actually used during the destruction of um, the foreign bodies as well as the damaged organelles are the functionless, useless, senile organelles of the inside the cell. So if we talk about the size and shape, the size of the lysosome that range from the 250 to 750 nanometer, and they are spherical in shape. So the shape is the shape of the lysosome is spherical, and their size range from the 250 to 750. There are two types of the lysosome, the primary lysosome and the secondary lysosome. Now, what is the primary lysosome? These are the newly formed lysosome, and these that don't join the digestive events On the other hand, secondary lysosome, when primary lysosome contract and fuses to structure that contains substances which is to be digested, they are called as the secondary lysosome. So now we can see here this is the primary lysosome. It don't have the substance which is going to be digested by this lysosome. So this is known as the primary lysosome. But when there is fusion of this lysosome with the food or with the particle which is going to be digested by it, this combined fusion from this lysosome along with that particle to be digested is that is known as the secondary lysosome. And these primary lysosome, that is newly formed lysosome, they had been formed and manufactured by the released by the Golgi bodies, Golgi apparatus. So their main function is the lysosome is to the digestive system of the cell, and exogenous substances such as bacteria that are engulfed by the cell are digested by the lysosome. The lysosome contains the hydrolytic, the digestive enzyme for the digestion of different substances. They are engulfed by the lysosomes. So it's very important that which type of enzyme lysosome contains. So this is the hydrolytic enzymes. So you can see here there is a process which is known as the phagocytosis or the cell eating. Uh, there is some extra particle that is being entered into the that cytoplasm that is being enclosed by a vesicle, and these particles, suppose if they are the food particles, they are enclosed in the vesicle. That vesicle is known as the food vesicle. Then there is attachment of the lysosome with this uh, food particles at the food vesicle, and the lysosome then digest or destroy if it is a foreign body. Similarly, if there is old, which means senile or damaged organelle, like we can see here, this is a mitochondria that is being damaged, so it is being attacked by the lysosome, and that lysosome is being released by the Golgi apparatus, both for the digestion of the phagocytized food particles of the cell and for the old and damaged organelles that is being attached, and hence the breakdown of the old organelles and recycling of the old organelles. Second function is the excretion. Lysosome remove waste material from cell. The waste material may be the waste of digested food or any damaged cell part. The third function of the lysosome is the antibacterial action. Lysosome contain antibacterial enzyme including the lysozyme, which digests the bacterial cell membrane, and the lysophilin, which binds with iron and other material and acid at the 5 pH, which activate the hydrolysis. So this is actually the process which we have discussed. This is the bacterium which enter into the cytoplasm through the phagocytosis process that is being enclosed by a vesicle, which is known as the phagosome, and then that phagosome is then attached and accompanied by the lysosome. Lysosome and phagosome they combine together, and the bacteria inside that phagosome that is being digested, destroyed, as we can see here. So this is actually the function of the lysosome, and this cell is actually a macrophage cell. The another organelles which is known as the peroxisome, which are similar in structure, but they are smaller in size than the lysosome. They are different from lysosome in two ways. They are believed to be formed by the self-replication rather than formed from the Golgi apparatus. The lysosome, they are released and formed by the Golgi apparatus, while these are self-replication structures. The second difference is that they contain the oxidase enzyme. As we discussed in the previous slide, that the lysosome contains hydrolysis enzyme, while this one, this peroxisome, they contain the oxidase enzyme. These oxidases enzyme they combine oxygen with the hydrogen to form hydrogen peroxide, and then hydrogen peroxide in combination with catalase, which is an oxidize many substances that might otherwise be poisonous to the cell. So this is actually the reaction where this catalase. So this is actually the formation of that hydrogen peroxide. Then H2O2, a catalase, they will convert them into the oxygen and water. This hydrogen peroxide, they may combine with some structure H2, and then it will be converted into the same 2H2, and that structure is which is being attached with the hydrogen, they will be detached. So the oxidation of fatty acid is accompanied by the production of hydrogen peroxide from oxygen. The hydrogen peroxide is decomposed by catalase either by conversion to water or by oxidation of another organic compound, which is being designated here by the AH2. So this is the structure of the cell. This is the nucleus. This is the nucleolus. This is the Golgi bodies. These both are the mitochondria. These are the cytoskeleton or the tubulin system of this the cytoplasm or the cell. This green line, this is the cell membrane. These are the endoplasmic reticulum. These are the lysosome, and this lipid part is known as the cytosol. Thank you so much to all of you. That was the third lecture of cell and its function. Hope there will be no such problem. All the best for your upcoming exams. Thank you so much. Allah Hafiz.